Hello, my name is Dariusz Kulus. I'm from the Bydgoszcz University of Science and Technology in Poland, and it is my great pleasure to be with you here today during this wonderful conference. And during my presentation, I would like to tell you a few words about the application of nanoparticles in horticultural biotechnology of ornamental plants. So basically, nanoparticles, due to their unique properties, as you know, can be used in various scientific disciplines, such as material science, technology, chemistry, physics, but also in life sciences, such as biology, medicine, and also in agriculture. For example, nanoparticles have been used for agricultural purposes as fertilizers, as herbicides or fungicides, also as nanosensors, and as carriers of gene transfers in the genetic engineering programs. And in our work, we have focused on the use of nanoparticles in plant biotechnology, and we have decided to combine nanoparticles with plant tissue cultures. Plant tissue cultures or in vitro cultures are the most basic tool used by biotechnologists. This technology is based on the cultivation of plants on synthetic media, such as those that you can see here in the photograph, and such plants are grown in controlled growth conditions, and tissue cultures can be used for various purposes. For example, for the purpose of large-scale reproduction of plants, for breeding, the creation of new cultivars, and the protection of biodiversity, but they can be also used for other reasons, for example, for the production of secondary metabolites. And in our study, we have decided to see how the addition of nanomaterials, nanoparticles of various metals into the culture medium affects the efficiency of these tissue culture techniques, and how do nanoparticles affect the development of plants. So, in the first part of our experiments, we have verified the utility of nanoparticles, different types of nanoparticles in micropropagation. Micropropagation is a large scale method of plant production. As I said, it is performed in vitro by putting pieces of plants, the so-called explants, on the synthetic media. The media usually contain all the micronutrients, vitamins, and plant growth regulators that the plant needs to develop, to grow, and by manipulating with uh, the composition of this medium, we can stimulate the development of the explants. And here we were curious if the addition of nanoparticles can stimulate or perhaps inhibit the growth of such plants in vitro. Because depending on the conditions, depending on what type of chemical compounds we will add into the culture medium, we can stimulate different responses, different growth responses of plants. For example, we can stimulate either the development of shoots or roots or of somatic embryos. That depends on the goal. But since there is little information on the use of nanoparticles in plant tissue culture, we wanted to learn these effects. So Here you can see the impact of silver nanoparticles on the development of adventitious shoots in Chrysanthemum. In this study, we have inoculated some internode segments on the culture medium, and then directly after placing the explants on the medium, we have added some nanoparticles onto the surface of the medium. In this study, we have tested silver nanoparticles at different concentrations, 50 or 100 ppms. And as you can see in this particular species and this explant type, the addition of nanoparticles was not very successful. The development of plants was clearly inhibited in comparison with the non-treated control. However, you should keep in mind that the final effect of plant tissue culture depends a lot on the genotype, on the species, sometimes even on the cultivar of the plants, and also, of course, on the type of nanomaterials used. And on the other hand, with bleeding heart, we have also studied the effect of nanoparticles on the efficiency of plant development, but this time we used gold nanoparticles. And here, in this species, you can clearly see that the addition of gold nanoparticles at the concentration of 75 ppms stimulated the development of plantlets in comparison with the control. So here the effect was very positive, and with this species gold nanoparticles could be substituted instead of traditionally used plant growth regulators. But as I said, the efficiency of micropropagation depends on many factors, also on the cultivar type and on the explant used. In these figures, we can see the effect of different types of nanomaterials. This time it were silver nanoparticles, but also zinc, the zinc oxide nanoparticles, and a mixture of zinc oxide nanoparticles and silver nanoparticles. And again, those nanomaterials were placed on the surface of the culture medium, but this time we also used chrysanthemums. However, we used different cultivars, and we have inoculated leaf segments on the medium. And here the results were also very interesting because, as you can see, in chrysanthemum pinky gold, where basically there were no differences between the different types of nanoparticles used in terms of shoot development, the dynamics of shoot development, the number of shoots developing on the explants. But as for the cultivar Burgundy Gold, here we observed a highly stimulating effect of nanoparticles on the development of adventitious shoots. Because as you can see, in the control object, there was basically no shoot development observed. In the control object, there were no nanoparticles, of course, or no plant growth regulators in the medium. But as for the other treatments, where the materials were placed on the culture medium, we observed that the plants developed more intensively. So this was also very promising for further research. But basically, here you can clearly see how different types of nanoparticles affect in a various way the development of various cultivars or even explant types. So basically, we need to optimize all of those parameters when developing a micropropagation protocol. We also observed some interesting effects of nanoparticles on the ex vitro growth of the plants. Ex vitro means that after cultivating in a tissue culture in the jar, the plants are next transferred to pots, to a greenhouse, for example, and they are being acclimatized to natural conditions. And this is a very critical step because the plants that are growing in the tissue culture environment differ anatomically and physiologically from plants which are growing in normal outdoors conditions. Because due to the very high humidity in the culture vessel, the stomata of the plantlets growing in vitro are constantly open. They are dysfunctional. Moreover, because we are adding sugar into the culture medium, the plants which are growing in vitro are not photosynthesizing, they are not autotrophic, they are actually heterotrophic because they use the sugar that they are given in the culture medium. So the acclimatization of the plants from in vitro to in vivo conditions is a very important step that determines the success of micropropagation. So in order to enable the plants to survive in the ex vitro conditions, we need to take care of them for the first few weeks. For example, by watering them quite often to prevent them from excessive stress due to higher temperature, higher light intensity, and also higher temperature in comparison to the conditions in the growth room during the in vitro growth phase. And here we observed that if the plants are treated with gold nanoparticles before transplantation to ex vitro conditions, their survival is much higher in comparison with the control. Here you can see the effect of different treatments. We have studied here the effect of various chemical agents such as nanoparticles, but also physical agents on the survival and quality of the plants of bleeding heart transplanted to ex vitro conditions. And as I said, we observed that the survival of the plants that were treated with nanoparticles, gold nanoparticles, was much higher than of the control. And similarly, also the quality of the plants after 6 weeks of glasshouse growth was significantly improved if the plantlets were treated with gold nanoparticles. As you can see, most of the plantlets after acclimatization were of superb quality after the treatment with nanoparticles, gold nanoparticles, whereas less than 50% of control plants were of such high quality. So here we can also observe the positive impact of nanomaterials on the development of plants and the possibility of using nanomaterials in plant biotechnology, in micropropagation in particular. But plant biotechnology is not only about multiplication of plant material, it is also about breeding, creating new attractive cultivars. In my department, the Department of Ornamental Plants and Vegetable Crops, we are working mainly with horticultural plants, with ornamental plants, mostly with Chrysanthemum, which is one of the most popular ornamental plant species, and we have a long history of creating new cultivars of this economically but also culturally valuable plant species. In the past, for this purpose, for the purpose of mutation breeding, we used mainly physical agents such as gamma rays or X-rays. However, the access to such mutagens for breeders is very difficult because usually the machines that generate X-rays or gamma rays can be found in medical facilities or atomic agencies. And additionally, their use is dangerous for the humans because, as you know, X-rays or gamma rays well are mutagens basically. So you need to be extra cautious when using such factors. That's why we were interested to study if nanoparticles, which are much easier and cheaper to access than ionizing radiation, can be also effectively used as chemical mutagens, and if they can be used for the production of new cultivars of plants. For this purpose, we used silver nanoparticles applied at high concentrations, up to 100 ppms. And here we again tested the effect of nanoparticles on the genetic stability of chrysanthemum plants that were regenerated in the presence of those nanoparticles. And the results were also very interesting because we have observed again that the increasing concentration of nanoparticles reduced the development of adventitious shoots, but that's quite normal in mutation breeding because usually when you use a mutagen, it is a quite aggressive factor. So it affects the plant development negatively, but at the same time, it can alter its genetic structure due to the induction of some mutations. And here we also observed that the plants that were grown in the presence of high concentrations of silver nanoparticles after acclimatization to the greenhouse and brought to full flowering produced new interesting phenotypes. As you can see, this was the original cultivar, the Lilac Wonder cultivar, and after treating these plants with nanoparticles, we were able to produce plants with an altered plant color, inflorescence color, or even shape. And here you can see some examples of the chrysanthemum cultivars that were recently produced in our department. These cultivars received plant breeders' rights, and as you can see, they are very attractive due to their shape or due to the color. They are either burgundy gold or pinky gold. And this was a very interesting effect, especially with the burgundy colored flowers, because this color is quite rare in chrysanthemum, and it is highly valued by both producers and consumers. So we hope that we will be able to introduce those cultivars to the market. Especially that we are the only department, the only unit in Poland which produces our own national cultivars of chrysanthemum, because all others are imported from abroad. So, since nanoparticles affect the development of plant, its morphogenesis, and also affect the genetic stability of the plants, we were also interested whether nanoparticles can be used for the acquisition of secondary metabolites, because many plant species can be a source of valuable metabolites useful in medicine, in treatment of various diseases, bacterial, viral, or fungal diseases. So we tested the metabolite profile of plants treated again with different types of nanomaterials, of nanoparticles. There were many research like this performed in our laboratory. I will not tell you about all of them, just some of the most interesting. But basically, you can see clearly that depending on the type of nanomaterials and their concentration, they can affect the metabolism of the plant and the synthesis of both primary and secondary metabolites. And for example, the addition of gold nanoparticles stimulated the production of polyphenols and tannins in bleeding heart. Bleeding heart is an Asiatic plant species. It is an ornamental plant species, but also used in medicine due to the production of metabolites. So here we observe that by adding nanoparticles into the culture medium, we can stimulate, increase the production of those metabolites. So this can also be useful for the pharmaceutical industry or cosmetic industry, for example. And finally, our research focused also on the possibility of using nanoparticles in the preservation of biodiversity. Because recently we have faced a severe problem of genetic erosion. Genetic erosion means that in general the biodiversity on Earth is decreasing. And we need to protect this biodiversity for future generations, for example, for breeding purposes. And one of the most promising and successful methods of biodiversity protection is cryopreservation. Cryopreservation is a technology which is aimed at storing biological samples in liquid nitrogen at the temperature of minus 196 degrees. At such low temperatures, the metabolic activity, physiological activity of cells is arrested and they can be stored for very, very long time spans in such cryogenic conditions. However, as you can imagine, it is not so easy to just simply put a living cell in liquid nitrogen and keep it still viable. This is a quite complex process which requires different actions on the plant material, different treatments to prepare the plant for the stress of low temperature storage. And usually, to make a long story short, a cryopreservation protocol includes these following steps. First, the explants are pre-cultured on a medium with an increased osmotic pressure in order to stimulate some initial dehydration of the plant. Usually also abscisic acid is added at that step because this is a hormone that stimulates some stress responses in plants. Next, we are isolating the meristems, which are most suitable for cryopreservation. Then the meristems are encapsulated in sodium alginate. This alginate bed is an additional physical protection for the explant. Then the explant is dehydrated in a special solution, a mixture of so-called cryoprotectors, which are substances that should increase the stress tolerance of plants, and then the material can be stored in liquid nitrogen, and when needed, it can be again recovered in vitro. And in our studies, we have tested the effect of various types of nanoparticles, such as silver, gold, or zinc oxide nanoparticles that were added at various concentrations, either into the pre-culture medium, to the synthetic seed to the encapsulation alginate bead or to the recovery medium. And again, we observed some interesting effects because there was indeed a significant impact of nanoparticles on the survival of the stored biological material. However, this effect was dependent on the step of cryopreservation and also on the type of nanomaterials. So in general, the addition of nanomaterials, especially zinc or silver, into the agriculture medium was able to increase the survival of the explants in some of the tested cultivars, not in all of them, but some of them in fact had a higher survival. It was not a very big change, although it was still significant. It was somewhere about 10 or 15%. The addition of nanoparticles, especially of gold nanoparticles and zinc oxide nanoparticles into the capsule was particularly effective because the explants that were treated in such a way increased their survival after cryogenic storage from 20 to even 30%, and that was really a big improvement. On the other hand, the addition of nanoparticles into the recovery medium had a negative impact on the survival and recovery of the plant material, because we observed that the cell membranes of the plants that were explants that were inoculated after cryostorage on the medium with an addition of nanoparticles were disrupted. So the cells got damaged. Still, we were able to see that nanoparticles can be useful in cryopreservation if they are added into the alginate capsule. So basically, to summarize my speech, the effect of nanoparticles is strongly connected with their type, concentration, and method of plant treatment. Metal nanoparticles can be used alternatively to traditional growth regulators as they affect the morphogenesis of plants in vitro. However, this effect is species specific. Silver, gold, and zinc oxide nanoparticles applied at low concentrations during the encapsulation step of the cryopreservation protocol have a beneficial impact on the survival of the stored specimens without altering their DNA sequence, although again this positive effect may be cultivar specific. Conversely, the addition of nanoparticles into the recovery medium after rewarming of samples has an unfavorable impact resulting from the disruption of cell membranes and oxidative stress damage. We know that because we have studied the activity of various enzymes in the plants after treatment with nanoparticles. And finally, silver nanoparticles applied at high concentration are a cost-effective mutagen that can be useful in the creation of new cultivars of plants. And as I said, nanoparticles can also be used in the acquisition of secondary metabolites. Our research is supported by the National Science Center in Poland. We were awarded recently with 2 research projects. One of them about the use of nanoparticles in chrysanthemum breeding is now complete. The other about the use of nanoparticles in plant cryopreservation is still ongoing, so we will still do some more research on the impact of nanoparticles on the physiology, the stability, the ultrastructure of the plant material to learn the effect in a more precise, more detailed way. If you are interested in our research, I invite you to read some of our recent publications, which are also focused on the use of nanomaterials in plant biotechnology and plant tissue culture. And if you are interested in cooperation, we are always open and happy to cooperate. So you are always welcome to email us or contact us or check our website. So thank you very much for your kind attention. I hope that this lecture was interesting for you, that you were able to learn something new about different aspects of the use of nanoparticles in life sciences. And I wish you all the best and a pleasant conference. Thank you very much and goodbye.